The information in this module is accurate and complete to the best of our knowledge. All recommendations are made without guarantee on the part of the author or the sponsoring institutions. The author and the sponsoring institutions disclaim any liability in connection with the use of this information.
Welcome to the Next Generation Sequencing lecture. One of the main reasons for this lecture is that it is crucial to understand how the data is generated in order to design genome projects, and for the downstream analysis of the sequence data. The revolution in genomics is riding on the rapid development of next-generation sequencing technologies. In this lecture, we'll introduce the principles behind some of these approaches. At the end of this lecture, you should be able to mention the main advantages and disadvantages of the next-generation sequencing methods. Describe the Pyro Sequencing approach, the semiconductor approach, and the nanoball approach to the NGS. Describe the differences between the Emulsion PCR and bridge amplification. Explain the meaning of single read, paired end read, and mate pair. Mention the advantages of single molecule real-time sequencing. Describe the principle behind membrane sequencing. Recall the description of the Sanger sequencing from the first lecture. We'll refer to it as the first generation technology. The addition of the mixture of dNTPs and ddNTPs to the PCR resulted in the sequences that would randomly terminate at different lengths. These fragments can be subjected to electrophoresis and would align, forming a ladder with different colors, indicating a nucleotide present at the end of each sequence. A sequence is then read by a laser and converted to a computer file. The term next-generation sequencing, or simply NGS, refers to non-Sanger-based high-throughput DNA sequencing technologies. Most of the NGS involves sequencing by synthesis on a single short-strand template attached to a surface or bead. In the figure, you can see a slide with millions of dots representing attached fragments. Each fragment is carrying its own sequencing reaction, where a fluorescently labeled nucleotides are added one at a time. In each sequential cycle, a nucleotide is added and the resulting fluorescence is read by a laser, or a high-definition camera, or even a pH meter, depending on the technology used. NGS data is a lot cheaper, and NGS sequencers have a huge output, but it still has one obvious limitation compared to the Sanger standard. Specifically, the short reads are very short. Common output of the NGS technology is still at about 150 base pairs per read. Therefore, NGS data requires assembly, and this assembly is a lot more computer intensive. Usually, NGS assembly involves fragments of different lengths. The short reads are assembled into contigs, and the longer paired end reads and the mate pairs are used to join the contigs into larger assembled scaffolds, which may include some gaps in the locations where the contigs are not overlapping. The first very popular example of next-generation technology was a 454 sequencing by Roche, known as Pyro Sequencing. This method was based on a principle of sequencing by synthesis. In Pyro Sequencing, DNA fragments are first attached to the beads, clonally amplified inside emulsion bubbles in the process called Emulsion PCR. Then, Each bead is placed inside of a well on a microarray. These wells look very much like honey wells in a beehive. A camera is placed above the well and the first nucleotides is added. If the nucleotide is incorporated, it sets a chain reaction resulting in a pyrophosphate release and a flash of light. Hence the name Pyro Sequencing. If there is no flash, nothing is recorded. If the flash is twice as intensive, two identical nucleotides are incorporated. Thus, the Pyro Sequencing reaction consists of many cycles of incorporation. In other words, by monitoring the synthesis of the complementary DNA strand. 454 has been a leader in the NGS technologies for less than a decade, but despite putting out longer and longer sequences, at some point the short reads were as long as 800 base pairs. The high price was making it vulnerable to competition. At this point, 454 has been discontinued by the Roche company and other technologies are taking the lead. The influence of the 454 technology can today be seen in the semiconductor sequencing by ion Torrent PGM and ion proton based on very similar principles, except the nucleotide incorporation is based not on the release of light, but on change in the pH due to the release of the hydrogen atom after nucleotide incorporation. PGM was the first affordable NGS benchtop sequencer sold on the market for less than $50,000 US. Our own Caribbean Genome Center was started with the purchase of ion Torrent PGM. This machine was popular in the hospitals and was promoted by its parent company, Thermo Scientific, for clinical diagnostics of germline and somatic mutations. The previous technologies are limited by several factors. One of them is the use of Emulsion PCR, which is a complicated and error-prone process. Instead of beads in the Emulsion PCR, nanoball sequences uses rolling cycle replication to amplify small genomic fragments into nanoballs. The base order is determined when fluorescent probes bind to complementary DNA, and the probes are then ligated to anchor sequences bound to known sequences on a DNA template. Nanoball sequencing increases quality of calls, and the dense positioning of clusters on the slide results in more data. Illumina Sequencing is by far the most widely used sequencing technology of today. It is very robust and accurate due to revolutionary principle of bridge amplification. The Illumina machines use slides they call flow cells. Forward and reverse primers are attached to the bottom of each flow cell. The single-stranded fragments with ligated primers are randomly attached to the surface. Priming occurs as free ends of ligated fragments bow or bridge to a complementary primer on the surface of the flow cell. After a number of cycles, bridge amplification produces a cluster of fragments, each with the same nucleotide sequence at the top. This is why the synthesis begins. A camera will detect fluorescence not from one, but from hundreds of identical molecules in the cluster, amplifying the signal hundreds of times. The universal sequencing primer is then hybridized to the free end to initiate the sequencing by synthesis reaction, detected by a camera step by step. Each flow cell produces 100 to 200 million spatially separated clusters. Notice that not the whole length of the fragment needs to be sequenced. only the ends. This results in a special kind of read called paired end read with known length and known sequence at the end but not in the middle. Paired ends add a lot of flexibility to assemblies because the length of the fragment can be varied from about 150 to 1000 nucleotides. Paired end reads can be assembled into contigs, but they can also be used to join contigs together into scaffolds. Unfortunately, technology cannot produce paired end reads that are longer than 1000 base pairs. However, there is a clever trick called mate pairs that allows using sequences longer than 1 kb and up to 10 kb on an Illumina technology. To sequence the ends of fragments that are large, the DNA is being circularized. The ends are attached to each other in reverse. A mate pair is formed. The ends are now cut so the fragment becomes small enough to sequence by bridge amplification. Due to their length, mate pairs are very useful to connect contigs that are far apart into scaffolds. Reads, paired end reads, and different size of mate pairs are used to complete the genome projects. In essence, a modern genome project improves by decreasing the number of contigs, by increasing their lengths, by combining contigs into scaffolds of higher and higher lengths. Ideally, the longer are the sequencing reads, the faster is this process. There are technologies now that allow increasingly longer reads. Third generation sequencing. Single molecule real-time sequencing, or SMRT, has been developed by PacBio. This is a parallelized single molecule DNA sequencing method that allows sequencing of very long fragments of DNA. The crucial difference from Illumina methods is that here, instead of the DNA fragment, a single DNA polymerase enzyme is affixed to the bottom of the cell, to which a single molecule of DNA attaches as a template. Single molecule real-time sequencing utilizes a zero-mode wavelengths, ZMW. When a nucleotide is incorporated by the DNA polymerase, the fluorescent tag is cleaved off and diffuses out of the observation area of ZMW, which is a light is detected. PacBio allows sequences of 10 to 20 kb in length, but they still have the disadvantage for a higher price and lower quality. The latter one is compensated by overlaying many copies of PacBio sequences on top of each other. This increases quality, but increases the price tag as well. Alternatively, PacBio sequencing have been used to join the contigs in hybrid assemblies in combination with Illumina sequencing. Most recently, a new technology has been making headlines. This is called membrane sequencing and is represented here by the technology developed by a company called Oxford Nanopore. The sequencing is conducted as fragments of DNA are pushed through nanopores in the membrane, each nucleotide generating a specific detectable change in the current that flows across the membrane and is detected. One of the advantages of this approach is that it promises to be very cheap and so portable you could potentially do all the sequencing on a device attached to your laptop. So, in review, we looked at the recent strides of sequencing technology from the first generation Sanger to the next generation's technologies started with Pyro Sequencing, represented by 454, Ion Torrent, and Illumina Sequencing, which are the most common technologies today. In addition, we looked at the two of the third generation are single molecule sequencing technologies that will be more and more common in the coming years. This last slide has side-by-side comparison of the cost and output for the machines we have reviewed. Each of the genome projects has to be planned using cost and output estimates. De novo sequencing asks for higher coverage and longer reads, while resequencing projects may be accomplished with a limited amount of short reads. And don't forget that there is an error rate associated with all of these, and Sanger is still used as a golden standard in verification experiments. Here are some review questions following this lecture. This concludes today's lecture on the Genome Projects, the next generation sequencing. We will be seeing in the next lesson where we will study the topic of genome assembly.